Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on photosynthesis. In the previous video, we are already discussed about the definition of photosynthesis, the meaning of photosynthesis, the structure of chloroplast, and also pigments involved in the photosynthesis. In this video, we are going to discuss about utilization of light energy by photosynthetic pigments. Okay, so let's see. So how they are going to utilize the light energy? So the light energy falling on the plants, green plants, that is not completely absorbed by the plants. So it is reflected by them. Okay, only a little amount that passes through them. Only 1% of the light energy absorbed by the pigments. Only 1% of the light energy is absorbed by the pigments. Actually, they absorb visible spectrum. Okay, so let's see what is visible spectrum. So, the visible light is the portion of electromagnetic spectrum that the human eye can detect. Okay, so it ranges from the wavelength of 380 nanometers to the 700 nanometers. So actually this is the visible light. It ranges between 380 nanometers to the 700 nanometers. So it allows us to perceive the light. So actually this is the visible light. Okay, VIBGR. It consists VIBGR colors. Okay, so it's a combination of 7 colors, but it appears in a white color. Okay, violet, indigo, blue, green, yellow, orange, red. So it's a combination of colors, but it appears as a white light. Okay, it ranges between 380 to 700 nanometers. Plants are going to utilize this visible light only. Before this, ultraviolet rays are present. After this radiation, there are infrared rays are there. Okay, both are dangerous to us. So we are perceiving this visible light only. Plants are also going to utilize this visible light only. So this radiation is called as photosynthetically active radiation. Actually, plants absorb very little in the portion of green wavelength. Okay, they absorb very little amount of light. They reflect more amount of light in the green region. That's why the plants appear in green color. So the chiefly absorbed radiation is red and blue wavelength of light of the spectrum. Okay, so let's see what is absorption spectrum, what is action spectrum. So absorption spectrum is nothing but this is showing a graph plotted between wavelength of light and amount of light energy absorbed by the pigments. So here you can see the carotenoids, they absorb more amount of light in the green region. Whereas chlorophyll A shows the peak absorption in the blue region, whereas in the red region also. In the same condition is observed in chlorophyll B also. More amount of light is absorbed in the blue region as well as red region. Okay, whereas carotenoids, they absorb more amount of energy, more amount of radiation in the region of green region. Okay, so that's why carotenoids, they reflects these higher radiations. Okay, let's see the second one, that is action spectrum. So it is also a graph showing between different wavelengths and rate of photosynthesis. The graph is plotted between different wavelengths and the rate of photosynthesis. So it is difficult to measure the rate of photosynthesis. That's why you can measure this through the oxygen evolution also. If high amount of oxygen is released, that shows the high amount of photosynthetic rate. That's why he placed aerobic bacteria and also green algae Spirogyra. Okay, he made an experiment. Actually, this was performed by a scientist Engelmann. He performed an experiment to describe the action spectrum. So he placed aerobic bacteria media, so aerobic bacteria, they require high amount of oxygen. Okay, so when the green algae is subjected to the different wavelengths of light, in the case of blue region and also red region, more number of aerobic bacterial growth is observed. So it shows that more amount of photosynthetic rate is observed in the blue region as well as red region. So it concluded that the peak absorption rates are reported from red region and also blue region of the absorption spectrum. So let's see what is Hill reaction. Hill reaction is nothing but light driven splitting of water molecules in chloroplasts. Okay, so water is going to split into hydrogens, electrons and oxygen. So 2 protons, 2 electrons and 1 nascent oxygen will come. Okay, he wanted to prove that, Robert Hill wanted to prove that, so the oxygen is coming from the H2O, not from the CO2. So Robert Hooke took isolated chloroplasts. He exposed them to light in presence of an artificial hydrogen acceptor such as potassium ferric oxalate. So this is the actual reaction proved by Robert Hill. He took isolated chloroplasts. Okay, he took an hydrogen acceptor. Okay, so when he placed isolated chloroplasts in the light in presence of an artificial hydrogen acceptor, he got the reduced product. So you can say simply that is a glucose or a carbohydrate. Okay, so he got the reduced product, reported the release of oxygen, molecular oxygen. Okay, so A is nothing but hydrogen acceptor, 2AH2 is nothing but reduced product, and O2 is molecular oxygen that is released in this reaction. He proved that in absence of CO2 also there is evolution of oxygen. The same thing, the evolution of oxygen from water is reported by the 2 scientists such as Martin Kamen and Charles Rubin. They worked on sulfur bacteria. Okay, so they worked on sulfur bacteria. When the bacterial cells, they exposed to the light in presence of H2S, hydrogen sulfide, they took carbon dioxide, they fixed it into a carbohydrate form. Okay, so here there is no water. Instead of water, they have taken H2S. That's why instead of oxygen, there is a release of sulfur. Okay, so they proved that the sulfur released from H2S. Instead of water, they have taken H2S. Okay, they proved that oxygen is coming from water molecule, not from the CO2. Okay. Let's see the next important topic, that is Emerson Enhancement effect. So let's see what is Emerson Enhancement effect. So Emerson Enhancement effect is nothing but when red light and far red light. Red light ranges about 680 nanometers, whereas far red light ranges 700 nanometers. When they gave together to a photosynthesizing plant, the rate of photosynthesis is higher than the sum of their individual effects. Okay, so that was proved by Emerson. Robert Emerson worked on chlorella. When he gave higher wavelengths to the chlorella, so the rate of photosynthesis will be decreased gradually. So he observed that, so at the higher wavelengths of light, that decrease of photosynthetic rate at higher wavelengths is called as red drop. Then he made an experiment. He gave lower wavelengths as well as higher wavelengths simultaneously to the chlorella. He got observed that the photosynthetic rate is higher than the individual effects of the both wavelengths. He observed that the photosynthetic rate is drastically increased, that is higher than the individual rates of both the wavelengths. He observed that. Okay, so this leads to the discovery of photosystems. PS1 and PS2 were discovered basing on the Emerson Enhancement effect. So let's see light harvesting complexes. So light harvesting complexes are nothing but pigment protein molecules. They present in plants, algae, and also in cyanobacteria. Okay, so there are two photosystems, PS1 and PS2. Okay, so if you see the light harvesting system that contains two parts. They are reaction center and antenna. So middle chlorophyll A is present. So that is called reaction center. So reaction center is surrounded by so many pigment molecules, chlorophylls, xanthophylls, carotenoids. They surrounds the center chlorophyll A pigment. Center chlorophyll A pigment is nothing but that is nothing but reaction center, and these are the antenna. Okay, so they all capture the sunlight. They transverse the light to the reaction center. So then the reaction center converts the radiant energy into chemical energy. So this is one light harvesting system. Okay, so there are two light harvesting systems, PS1 and PS2. So PS1 nothing but the reaction center. In the reaction center, chlorophyll A that absorbs 700 nanometers wavelength of light. In case of PS2, chlorophyll A of reaction center that absorbs the wavelength of 680 nanometers. So that is the only the difference. So where do they located in plants? So already discussed in the previous session. So the pigment molecules such as xanthophylls, carotenoids, chlorophylls, they located on the membrane of thylakoid. If you see the membrane of thylakoid, you will find these photosynthetic subunits, nothing but quantosomes. Each quantosome is a light harvesting complex. Okay, so if you see the light harvesting complex, so it consists so many pigment molecules. That is called quantosome. If you see here, center is chlorophyll pigment that's surrounded by so many pigment molecules. So I have mentioned so many colors. Why? Because there are carotenoids, xanthophylls and chlorophylls. That's why I have mentioned different colors. Okay, so this is the actual photosynthetic unit, nothing but quantosome. Okay.